So this video is all about population ecology. This is really where we're starting with the major content of the course. As the course progresses, we're going to be moving up that ecological hierarchy from populations to communities to ecosystems and so on. So this video really covers chapter 10 of your textbooks, which is all about distribution and abundance of populations and species. So this lecture is going to be split into a couple of major themes. The first being defining what a population is, so that's clearly extremely important. Then we're going to talk about some of the factors that influence population distributions, density and abundance. And then finally we're going to talk about some of the methods that ecologists use to estimate species abundance or population sizes. So what is a population? Well, a population is a group of potentially interbreeding individuals of a single species inhabiting an area delimited by natural or human-imposed boundaries. So let's talk a little bit about what might constitute natural or human-imposed boundaries. On the picture on the left, you have a lake where obviously there are many organisms within that lake, the populations of which are limited by the magnitude of that lake, and there's very little dispersal of those organisms between that lake and other lakes. Equally, you've got a mountain range in the background with a tree line that's kind of stretching up the mountains, and at a certain point those trees are limited probably by low temperatures at high altitudes, and so that's another example of a kind of natural limit to the extent of a population. On the right hand side, this is an example of a human-imposed boundary. That's Central Park in New York, and obviously there are many species that occur and may be abundant within Central Park that don't stretch beyond the boundaries of that park and into the kind of dense urban areas that surround it. So why is population ecology important? Well, just very briefly, because it pertains to a lot of questions associated with things like conservation, so managing populations of endangered species, controlling pest populations. This is an outbreak of biting midges in Scotland that reach plague-like proportions at certain times of year. And finally, managing things like fish and game populations. So it comes down to things as basic as human food supply. So when we study populations, there are certain attributes of those populations that we can study effectively and compare between species, between regions, and so on. Those are the distribution of populations, the dispersal of individuals within and between populations, the density of individuals within populations, and the abundance of species. So the first one we're going to focus on is distribution. So the distribution of a population is the size, shape, and location of the area that population occupies. And there are a number of factors that limit population distributions, and they can broadly be divided into two categories. So the first is the physical environment, so things like temperature, rainfall, soil type. And the second thing is biotic interactions, so things like intra and interspecific competition that we'll come on to later in the course, predation and disease. So this is an example from your textbooks. It's an experiment by Joseph Connell that just shows really nicely some of those different factors that can affect population distributions. So these are two different species of barnacle colonizing part of the intertidal zone. It's on the coast of Scotland. And what Connell found was that those brown barnacles on the top tend to colonize this higher band on the shoreline, and the grey barnacles colonize the lower portion. So the difference fundamentally in the habitat is that as the tide rises and falls, that upper band where the brown barnacles are is exposed to air for a much longer period of time. So the theory goes that the brown barnacles they're much more resistant to desiccation, so they're able to tolerate that dry environment. whereas the grey barnacles are limited to that lower portion of the beach because they can't tolerate desiccation to the same extent. But what Joseph Connell did was he transplanted some of those brown barnacles into the lower portion of the shoreline where the grey barnacles are, and he found that if he transplanted them there, they would survive there perfectly happily. And so what he realised was those brown barnacles were not limited, they were not excluded from the lower portion of the beach because of any physical factor of the environment. They were limited, or they were excluded from that portion purely by competition. So the distribution of the brown barnacles is restricted by competition with the grey barnacles, a biotic factor. And the distribution of the grey barnacles is limited by a physical factor of the environment, so their lack of tolerance for desiccation higher up the beach. Another element of that figure that you can see is the difference between fundamental niche and realised niche. So the fundamental niche of the brown barnacles is that whole stretch of beach. They could happily survive in any portion of the habitat, but they only happen to survive in that top portion of the habitat because they're restricted by competition with the grey barnacles. The grey barnacles' fundamental niche is just that lower portion of the beach because they're restricted from the upper portion by their lack of tolerance for desiccation. So their fundamental niche and their realised niche is actually the same. So like many questions in ecology, the distribution of populations look very different depending on the scale at which you're viewing them. And this slide talks about the distribution of individuals within a population on a relatively small scale. So those distributions tend to fall into three spatial categories. So random, which you can see on the left-hand side, a regular or even-spaced individuals that you can see in the middle, or a clumped distribution that you can see on the right. And these distributions of individuals tend to be determined by two major things. So social interactions between the individuals in the population and the structure of the physical environment, or a combination of the two. So if you look at those random distributions on the left-hand side, these usually come about in situations where individuals have neutral interactions with one another. So they might be basically ignoring each other. The regular distributions might occur when individuals have antagonistic interactions with one another. So it might be territorial animals that are controlling a particular space within that habitat. And then finally, clumped distributions occur when animals are attracted or organisms are attracted to one another. So for example, in social animals. In terms of elements of the physical environment, random and regular distributions might come about if there's a relatively even distribution of resources, or in terms of, particularly in the case of regular distributions, if there's generally a shortage of resources across the landscape, and so individuals are spreading out in order to maximize their utility of that resource. And finally, clumped distributions might occur because some resource, like water or food, that the animals require is itself clumped and not distributed evenly across the environment. When we look at population distributions at larger and larger scales, they almost always begin to appear clumped. And that's because on large scales we see increased levels of environmental variation. So for example, this is a map that shows the global distribution of coral reefs. And clearly they stick to the coastlines or just off the coastlines of tropical regions where there are shallow oceans that create habitats that can support corals. And so clearly when we increase the scale on which we're looking at things, just by the distribution of areas that can support species, we're going to see clumped species distributions. So the second feature of populations that we're going to talk about is dispersal. So what is dispersal? Well, it's different from migration. Migration is a seasonal movement of individuals from one location to another. I mean, it almost always involves a return movement, whereas dispersal is typically a permanent shift of an individual or a population to a new location. So several factors drive dispersal: the expansion of populations, range changes in response to climate change, and dispersal in response to changing food supply. So I'll go with some examples of each of those drivers. So that first driver of dispersal is expanding populations. So this tends to happen when a species is introduced either deliberately or accidentally to a new area, and we see gradual expansion of that population across a range. A good example of this is the introduction of Africanized bees to Brazil. So honey producers in Brazil wanted bees that they thought would be better suited to their warm climate. And so they introduced an Africanized subspecies of the honeybee, Apis mellifera, to Brazil. Because the Africanized bees come from an area where there are a higher number of nest predators, those Africanized bees exhibit much more aggressive behavior. And another thing they do more often is that they create swarms that then move to new sites and produce new nests. And so their colonies bud off and produce new colonies at a much higher rate than the European bees. So what we've seen ever since their introduction to Brazil in the 1950s is this incredibly rapid range expansion of that species, both south and north, which has spread all the way to kind of the middle of North America now. And it's only going to be stopped by colder temperatures in the northern part of North America. So the range of these Africanized bees is reportedly increasing by about 500 meters a year. And the other thing about Africanized bees is because they're so aggressive and because they sting humans on such a regular basis, that range expansion has been very easy to notice because people in their new range very quickly notice the increase in rates of bee stings. The second driver of dispersal is range changes in response to climate change. So a good example of that would be along elevation gradients. So you can see this mountain range on the right. You see a really distinct tree line at a certain point on those mountains, and that's because higher than that tree line, the temperature is too cold for those trees to maintain a population. But as temperatures rise in response to climate change, the line on those mountains at which the trees can no longer survive is going to get higher and higher. So as the temperature warms, we're going to see that tree line rising up the mountains. Now the worrying thing is for some of the small plants and lichens that exist only above that tree line. So their population range is going to be decreased, is going to be ever decreased in response to this change in the tree line. So the third driver for dispersal of populations is a response to changing food supply. So the example you can see on the right now is the relationship between voles and kestrels, so predatory birds, in some sites in western Finland. So the graph on the bottom shows these massive variations in populations of both voles and kestrels over time. But voles are very kind of short-lived mammals with rapid reproduction. And so where you see these huge increases in populations of voles, that's because we're seeing just huge increases in reproductive rates of the voles, probably because of climatic factors and the availability of food. But you can see that the kestrel populations follow the vole populations extremely closely, and kestrels are much longer lived and slower reproducing animals. So there's no way that the kestrels could follow the population patterns of the voles through changes in reproductive rate. So the only way that kestrels are able to follow the density patterns of the voles is through dispersal or immigration and emigration. And so what's happening is every so often we're getting huge booms in the vole population, and lots of kestrels are essentially coming into those sites to feed on the voles and take advantage of that increased food supply. So while we're talking about dispersal, it's a good time to introduce you to the concept of metapopulations. So populations may not always occur in a single continuous area, but often occur in spatially isolated patches with exchange of individuals occurring between them. So this is a metapopulation. So we'll probably talk later in the course about differences in rates of dispersal from large and small patches, etc. But for now, we're just looking at a couple of examples of a metapopulation. So these metapopulations might occur because subpopulations occur naturally in areas like groups of islands. These are mangrove islands that you can see on the left. But those separated subpopulations might also occur because of elements of kind of human disturbance. So you can see these fragmented forest fragments in the picture on the right. They're all kind of separated by farmland. So in either case, what distinguishes these metapopulations from just a group of isolated populations is that there's distribution or dispersal of individuals between those patches. So the next feature of populations I want to talk about is population density. So population density is simply the number of individuals per unit area. But there are two different ways, slightly different ways that we hear ecologists describing population density. The first is absolute density, so this is just the number of individuals per unit area. And the second is ecological density. So ecological density incorporates the idea of the niche of the organism that we're studying. So it's the number of individuals per unit area of suitable habitat. So this is useful because in certain cases, for example, if we looked at the mountain gorilla in Africa, these gorillas only occur in two very small areas of montane tropical forests towards the interior of Africa. So it might not be very useful to quote their density over the entire continent of Africa, when really their density is much more relevant over small areas where they can at least potentially occur. So this is a figure from your textbooks, and it shows this really well preserved negative relationship between the size of organisms and their population density. So essentially, as animals get larger the species, the lower the population density they tend to be found in. And in this figure, you can see that relationship broken down into different groups of organisms. So you can see that relationship holds true across these different groups, but there are sometimes some differences between groups. So for example, in large birds and large mammals tend to occur in slightly higher population density than large birds do. But across the board, there is still this really well kept negative relationship between organism size and population density. So the final feature of populations that I'm going to talk about is abundance. So firstly, what influences the commonness or rarity of species? Well, there tend to be three major factors that influence commonness and rarity. So geographical range. Is it high or low? Do species occur over a wide area of the planet, or are they restricted to very small areas? Habitat tolerance. So we saw this in Joseph Connell's barnacles. One species was much more tolerant of desiccation than another, and so that's likely overall to make that species more common. And then localized population sizes. So where species occur, do they occur in high populations or in low populations? So each of those three factors can be high or low, and where they're low, it tends to confer rarity in that species. What we find is that common species are rare, and rare species are common. So there are lots and lots of species that don't occur in particularly high numbers, and not very many species that we see really, really commonly. So why is that? Well, Deborah Rauwnaerts in 1981 came up with this classification of commonness and rarity. So 7 forms of rarity and 1 of abundance. And essentially what she was saying is that if you look at those 3 different factors, geographic range, habitat tolerance, and localized population sizes, there are 7 different combinations where at least one of those is low. and only one combination where all three of them are high. So we can quickly have a look at some examples of species with different combinations of these factors. So for example, let's look at a species with high geographic range, high habitat tolerance, but very low localized population sizes. So one example would be the peregrine falcon, which very, very nearly went extinct a couple of decades ago, despite having high geographic range and high habitat tolerance. So this bird, its range is most of the northern hemisphere. It has really high habitat tolerance, but its population sizes in localized areas tend to be very, very low. And so some kind of factor that influences those populations even slightly, like the use of DDT, can drive that species almost to extinction, and that's in fact what happened. So let's have a look now at a species with low geographic range, low habitat tolerance, and low localized population sizes. So clearly being low in all of these factors is going to make species extremely susceptible to extinction. And one example I can think of is the mountain gorilla. So this is a species with incredibly low geographic range. This is its range here. It's restricted to two tiny regions of Uganda and Rwanda. And within those regions, it has low habitat tolerance. So it requires pristine tropical forests that occur in kind of lower and lower areas as a result of deforestation and habitat loss. And even in those habitats where they do exist, they exist at very low localized population sizes. And that's because they're huge animals. They require huge levels of resources in order to kind of maintain themselves. And so having a low in all of these factors means these animals, as I say, are very susceptible to extinction. So I want to finish this lecture by talking about some techniques that ecologists use to estimate abundance of organisms. So the abundance of organisms and how that abundance changes over time and space are among the most fundamental concerns in ecology. And we use a number of different tools to estimate abundance. One would be the use of quadrats that you can see in this image here. And these are really helpful, but particularly for studying small organisms that obviously fit within manageable quadrats, and equally sessile organisms, so organisms that aren't moving around too quickly. In some cases, instead of looking at abundance as the number of individuals in a quadrat, we would look at estimating species coverage. So that's really useful for studying really small organisms or organisms where identifying an individual might be difficult. So for example, in this quadrat, you would estimate grass coverage as being roughly 100%, but it would be very, very difficult to determine how many individual plants there are in that quadrat. So another method we use for estimating abundance is mark and recapture. So this is really useful for studying populations of mobile organisms that we can't count in a quadrat, and particularly for very elusive animals where it's going to be really difficult to directly count the number of individuals. So this is the equation for mark and recapture. So capital N is the estimated total population size, so that's what you're looking for. Capital M is the number of individuals marked and released. And then during a second sampling period, small n is the number of individuals captured, and small m is the number of marked individuals that were recaptured in that second sample. So in some cases, mark and recapture doesn't necessarily involve actually marking individuals. So this is an example of the use of photo identification to estimate the populations of humpback whales in Frederick Sound, Alaska. And so what scientists do is they look at whales as they surface and identify distinctive marks on individuals. And so they can identify individuals that way. So in this case, over an initial sampling period, 72 whales were photographed and identified. Then during a second sampling period, 78 whales were photographed. And of those 78, 22 were recaptures. So these were individuals that had been photographed during the initial sampling period as well. So to estimate population size of humpback whales, we then use this equation from the previous slide. So this is 72 times 78 divided by 22, which gives us an estimated population size of these whales of 255.